Hi everyone, my name is Clorice Reinhardt and I am a 2023 Arnold Beckman Postdoctoral Fellow. Arnold Beckman was an incredible scientist, also from the Midwest, where his discoveries and inventions like the acidometer, the precursor to our modern pH meter, and the UV spectrophotometer have proven crucial for our ability to characterize and analyze the chemical composition of both biological and material samples. It is hard for me to think of a chemistry laboratory or an industry that is fueled by basic science, such as energy, defense, and commerce, that hasn't been profoundly impacted by his discoveries. Not unsimilar from Arnold Beckman, I too am from a small town in the Midwest, and I was inspired by my love of science to pursue a PhD. As a scientist, I am fascinated by understanding how the machines in our bodies work. In particularly, chemistry allows me to understand how chemical reactions happen and what controls them. So many important processes that we rely on for life, like photosynthesis, cellular respiration, and even nitrogen fixation by legumes in the soil, are only possible because enzymes can take really slow, sluggish reactions and make them faster by thousands of millions times to be compatible with the speeds that we need for life. They do this by making sure the reactant molecules are in the proper orientation and really just ensuring that everything goes to completion. So enzymes are great, but how do we take such powerful machines in the lab and tune them to really accelerate reactions that we want to happen in the conditions that we want them to? I would argue that we can't recreate what we don't understand. So just like a mechanic would take apart a car or a machine to understand how it works, I take apart enzymes and figure out how they work. My tool of choice, however, is the computer. So I blend physical chemistry and biochemistry to use tools from theoretical chemistry to model enzyme reactions, analyze large data sets, and really develop tools so that we can use these to understand how enzymes work and enable us to engineer new enzymes. So today I want to tell you a little bit about my work on metalloenzymes with the Beckman Foundation and how I was able to leverage their support in finding my independent career and really being part of such a great community. So a little bit about what I work on. So I'm interested in metalloenzymes. So these are nature's catalysts that have metals in the active site. So that could be iron, that could be nickel, manganese, all of these metals that help catalyze really difficult reactions. In particular, enzymes that use iron are of interest to me. They can have very similar active sites, but yet we know from previous scientific work that they can catalyze very different reactions. So it's clear that the effect of the environment beyond just the area immediately around the metal has a really important role in dictating what the enzyme can kind of accelerate, what the products are. And those are important knobs that we need to know how to tune in order to enable the chemistry that we want. So I'm really focused on trying to understand how we can kind of unlock those broad trends. To do that, I needed to construct a computational data set. So based on many years of scientific researchers before me, some may say that you stand on the shoulder of giants. That's certainly true. I was able to construct a data set of experimental structures stored in the protein data bank. So I was able to pull out all of the unique non-heme iron enzymes in the protein data bank, and this would serve as our data set to understand how these enzymes work and kind of disentangle the effects by the local environment on catalysis. Unfortunately, so as part of a computationalist, we need to be able to create reasonable physical models. And because these systems are so complicated, there may be, thousands of atoms, if not sometimes hundreds of thousands in our simulations, our calculations. We need to find a way to kind of automate that and make sure that it's done accurately. So we created a tool called Quantum PDB, which you can take your existing crystal structure or your experimental data, feed that into Quantum PDB. It will repair everything that's kind of missing about the structure, take out things you don't want, build intuitive models, and then prepare that for a quantum mechanical calculation where we can actually kind of see what the electrons are doing. And so this was something that not only works on metalloenzymes, but works on different types of proteins, and it's easily customizable. So we hope that this tool can help enable further studies. As part of this, gathering this data set and constructing these tools, we were able to kind of obtain a sense of, some really cool enzymes and proteins that hadn't been studied or weren't understood how they work. And so one of them that came out of there is called DMFase. So this is an enzyme that breaks an amide bond, so a bond between this nitrogen and carbon in red here, in the organic solvent N,N-dimethylformitamide. So this is a molecule that's typically used by the tanning industry or to make leather. And so it's something that can be, found in groundwater as a pollutant, especially in countries that produce a lot of leather. And so this is an enzyme that actually bacteria produce, that we discovered about, I'd say 40 years ago, that actually can cleave this CN bond, and the bacteria actually then use these molecules in their metabolism. And so we wanted to understand how this enzyme worked. And something that we did is we not only blended classical physics, so kind of understanding how this enzyme would move using kind of a ball and spring model, but also understanding how the reaction mechanism works using quantum mechanics. And then I also leverage bioinformatics, so using kind of existing genomic and sequencing data to really understand how prevalent this enzyme is, and how kind of conserved this active site is. So can other enzymes do similar reactions, or do they look similar? And you can actually see this in action. So this is the reaction happening. First, we form a ferric hydroxide. This is going to attack the molecule. You're going to have this part of the protein called a glutamate that's going to kind of move some protons back and forth there. And you're finally going to form the product at the end. And so this is something that with computation we can see happening, and we can understand if it's energetically favorable, and how we would kind of try to recreate that in a laboratory environment. And so beyond that, I don't want to take too much of your time today, but from that, I was also able to do some work on enzymes that have multiple irons, and what kind of cool chemistry can they do. So one of a big challenge in the chemical field in general is activating very kind of strong CH bonds. And what I mean by that is that some chemical bonds aren't inherently that reactive, and it takes a lot of energy to break those and replace it with something else that we want. So a particular transformation that we're interested in is taking, say, like the CH bond in methane and converting it to methanol, or the CH bond in octane, or another petrochemical, and converting it to something more useful. So one enzyme that does that is actually a bacterial enzyme called alkane monooxygenase. And so it actually converts octane or other medium to linear alkanes. Basically what that means is chemicals in oil. So this bacteria enzyme actually eats oil in some sense. And it converts it to a product that the bacteria use and they can grow on. So we were trying to understand how this mechanism worked. And this is something that I actually was able to do with other Beckman's recipients. So this was done with two Beckman Scholar undergrads in my collaborator, Professor Rachel Austin's lab, as well as her collaborator, Chris Pollock. And so this was something where we got to combine spectroscopy methods, molecular dynamics simulations done by me, and experiments done by mainly undergraduate researchers to really put together the pieces of how this enzyme worked. And this is something that wouldn't have been possible without the connections in the Beckman Foundation, as I wouldn't have had a chance to be introduced to my collaborator and her wonderful students. And so this is a kind of a rare example of where, not only Beckman funded postdocs can interact with a younger generation of undergraduate researchers, and really work together to tackle some of the hard problems out there, I guess. And with that, I'd like to thank my lab, but mostly the Beckman Foundation for really providing me financial support these last couple of years to tackle things that I'm really passionate about. I've had a chance to train some wonderful undergraduate researchers and interact with my whole colleague of postdoctoral fellows at the Beckman Conference, as well as meet, Tierney and Juliet of the Beckman Scholar Program, my collaborator, Rachel. And so with that, I'd like to thank you again for your support over the last couple of years, and for welcoming me as part of a really caring community of scientists that kind of allows us to chase the big dreams and goals. And so with that, thank you so much.